My name is John Chaput, and I'm a professor here in the School of Pharmacy and Pharmaceutical Sciences at UCI. As part of our homecoming activities, I was asked to say a few words about the COVID research that is ongoing in my laboratory. As I'm sure all of you are aware, the current coronavirus pandemic represents the greatest threat to U.S. health and prosperity since the Great Depression. While vaccines are on everyone's mind, it's important to remember that we have two other arrows in our quiver. The first is diagnostics. With more than 2.2 million deaths reported worldwide, 450,000 of which have occurred here in the United States, new technologies are urgently needed for routine, large-scale testing. Inexpensive and simple-to-use tests would control the spread of COVID-19, and importantly, they would facilitate safe environments for social and economic activities that include work, travel, and school. Currently, quantitative real-time PCR is the gold standard for detecting the SARS-CoV-2 virus in patients. Unfortunately, this method has slow turnaround times, generally 24 hours, and cannot be scaled to the entire population. Consequently, what we need most are simple tests that any person can use at any time to make an informed decision. I refer to these tests as point-of-need diagnostics, as they would be performed in the comfort of your own home when you need them, as opposed to, say, point-of-care diagnostics, which are performed in a doctor's office. With this goal in mind, my lab has developed a highly sensitive COVID-19 test that can give a result in less than 1 hour. We believe that this test and others like it are a major breakthrough in COVID-19 testing. Our idea is to make this test broadly available so that healthcare decisions can be made almost instantaneously by the patient. Patients that do receive a positive test would then have that result independently validated using the gold standard of quantitative real-time PCR. The second arrow in our quiver is therapeutics. My lab is also working to develop COVID-19 therapeutics. We are discovering synthetic antibodies that would block the virus from interacting with the host cell receptor. Although this work is still very much at the early stages of development, we have already discovered affinity reagents that can compete with the best human monoclonal antibodies. While drug discovery is a slow process, the pipeline that we are creating will ultimately allow us to respond much more quickly to future public health threats and help prevent local epidemics from becoming global pandemics. I appreciate you taking the time to listen to this research overview, and I hope that you and your families remain safe. Anyone interested in learning more about my research is welcome to contact me directly. My information can be found on the website for the School of Pharmacy and Pharmaceutical Sciences at UCI. Thank you and goodbye.
My name is Youngji Kwon, Professor of Pharmaceutical Sciences, School of Pharmacy and Pharmaceutical Sciences at UCI. Efficient vaccination requires successful and efficient completion of vaccine preparation, giving it to a patient, usually via intramuscular injection, moving of antigenic materials in the body to where immune cells reside while avoiding sequestration. And the most importantly, being eaten by key immune controller cells called antigen-presenting cells that present the antigen signals to start a cascade of antibody generation and the eradication of pathogen-infected cells. For nucleic acid-based and viral vector-based COVID-19 vaccines developed by Pfizer, Moderna, and AstraZeneca require the expression of the encoded SARS-CoV-2 viral proteins before the move and eat and present steps. Current COVID-19 vaccines have limited storage and distribution. The majority of the vaccine materials are not going to be utilized for vaccination, but wasted during the processes mentioned earlier. The immune stimulation supplement component, called adjuvant, could also provoke allergenic reactions. And the immune system might remain at an activated stage only for a limited period of time or often rapidly tamed when effector cells get exhausted in the body. To address these challenges, we have developed a novel technology that creates about 100 hundreds to thousands of small vesicles from each optimally programmed immune controller cell for immune activation against SARS-CoV-2 virus, preparing avatars, drones, or satellites, as we call them exchangeably. The resulting vesicles bypass the move step in vaccination and are already fully equipped for immune activation like their parent cells for the best eat and present step. Unlike the live immune control cells, the vesicle vaccine does not raise the concerns about administering live cells, but still performs like live cells. That's why we call them cell-free cellular vaccine. In addition, the vesicle vaccines can be further tuned to target a specific viral proteins, such as mutant spike proteins, and they can be completely personalized or universally applicable, depending on using an autologous or allogeneic cell source. We originally came up with this technology for cancer vaccination, and the very clear and exciting evidence of efficient protection of animals from tumors was demonstrated. In response to the unprecedented pandemic situation, we revamped the technology to make it a new class of COVID-19 vaccine. We successfully prepared the first version of the vesicle COVID-19 vaccine by genetically engineering immune controller cells, followed by converting the cells to micro-scale vesicles. A group of animals have been vaccinated with both prime and booster shots of the vaccine, and the subsequent serology studies in a few weeks will confirm and quantify the generation of SARS-CoV-2 neutralizing antibodies. Then the vaccine's prevention capability will be holistically assessed by using an animal model that mimic human COVID-19 pathology. This study was generously supported by the pilot funding at UCI, orchestrated by the Office of Research, School of Medicine, and School of Pharmacy and Pharmaceutical Sciences.
Hello, everyone. My name is Wei-An Zhao. I am a professor at the School of Pharmacy and Pharmaceutical Sciences, UC Irvine. I'm very excited here today to tell you about our COVID-19 serology test that can have very important applications in COVID surveillance and therapeutic and vaccine development. This is a work in collaboration with Dr. Philip Filner in the UCI Vaccine R&D Center. This test is to measure antibody response to see if you had COVID. We start with 1 drop of blood sample. Typically, we collect the sample through a finger stick, which is very easy and quick. And this can be collected, under this the drive-through setting as well. So then we add the sample to a microarray chip, which comprise a comprehensive panel of respiratory viral antigens. So these antigens are molecules that are found on viruses. So we can use them to capture antibodies in the sample. Using a salvage assay, we can tag this target antibody to produce fluorescence, which can then be measured using a portable imager we developed specifically for the point-of-care use. So effectively, we can run a couple of hundreds of tests on this single chip with the size of thumbnail. Our test has exceptionally high sensitivity and specificity, and it can outperform conventional method because of the large number of antigen we used in this assay. So this test is also very modular. As the virus continue to evolve, we can easily customize the antigen panel to accommodate new variants. So one of the most important applications of this test is to measure the prevalence of COVID infections for the public health surveillance. In collaboration with UCI School of Public Health and UCI Medical Center, we actually tested over 10,000 healthcare workers from UCI Medical Center and Orange County residents. Back in last summer, we found out about 10% of OC residents were seropositive, which is 7 times higher than what we initially expected. Over the past couple of months, you can see positive cases rise very rapidly. And by December last year, about 40% OC residents, especially in the Santa Ana area, are seropositive. Now, you also notice the exposure levels at UCI Medical Center campus is actually much, much lower than the OC and the Santa Ana area. This might be due to transmission control measures enforced by the hospital compared to those in the Santa Ana, in the community area. So these results support the use of transmission control measures, things like wear a mask, social distancing, wash hands to effectively limit the spread of virus. The other very important application of this test is to measure the efficacy of vaccines. We already have 2 vaccines on the market, and a few more are coming. So people want to know how well they actually work, which can be determined, with our test. So another exciting project we're working on right now is to develop a comprehensive viral antigen test that can diagnose not only COVID-19 infection, but also flu and common cold in a single test. This will allow us to effectively manage and treat patients with overlapping symptoms. Finally, my lab is also working on to develop super broad neutralizing antibodies for COVID using a very powerful single cell-based high throughput screening. We're particularly interested in the antibodies that can target multiple SARS variants, including the recent ones found in UK and South Africa. So once developed, these super neutralizing antibodies can be used as therapeutics or vectorized vaccines to treat and prevent COVID. So we're really excited about this technology. And I think what we're going to do in the next couple of months is to really scale up and automate this whole system so we can really deploy that very quickly to the clinical testing centers so we can really benefit more broadly in the society. And also we're very actively looking for partnerships with pharmaceutical companies. Like I mentioned, one of the things we're really interested in is to use these tests to measure the efficacy and how long those efficacy can last as well. For this vaccine candidate. So that's something we're really excited about to do in the next couple of months. Thank you so much. And if you have more questions, please feel free to reach out to me.